Hi everyone, my name is Irene Soler-Sáez and I'm going to explain the research done to unravel sex differences in multiple sclerosis implementing an integrated single-cell transcriptomic approach. It corresponds to my master's thesis and it was done in the Bioinformatics and Biostatistics unit of the Príncipe Felipe Research Center in Valencia, Spain. First of all, multiple sclerosis is an autoimmune and neurodegenerative disease of the central nervous system. It is characterized by the destruction of the layer that recovers neuronal axons. There is a sex bias in different epidemiological and clinical aspects of multiple sclerosis. For instance, its prevalence is higher in women, whereas the deterioration process is faster and more severe in men. Sex chromosomes and sex hormones are being evaluated to explain those differences. However, nowadays the molecular reasons that could assess them are poorly understood. For this reason, our aim is to describe the molecular mechanisms underlying sex-based differences in this disease. In particular, we develop an in silico approach analyzing single-cell RNA-seq and single-nuclear RNA-seq data through the following steps. Firstly, we performed a systematic review following the PRISMA statement to identify publicly available datasets. Secondly, each of the selected datasets was analyzed individually. This analysis included the download of the data and its preprocessing, the cell type annotation of each cell and nucleus evaluated, and two statistical analyses. In detail, we performed a differential abundance analysis to discover any significant difference in the proportions of cell types, and a differential gene expression analysis to identify different gene expression patterns that could reveal prospective biomarkers by sex for each cell type. Finally, we integrated the results from the latter among datasets and cell types. The common genes identified were characterized performing an overrepresentation analysis of the EU terms related with biological processes. As a result of the systematic review, we found 3 datasets that fulfill the inclusion criteria. 2 of them contain the single-cell RNA-seq data from blood samples, and the other one single-nuclear RNA-seq data from nervous tissue. The preprocessing step led us to work with high-quality data. Then, this data was used to assign the cell type to each cell and nucleus. In blood samples, we identified several types of lymphocytes, dendritic cells, and monocytes. In the nervous tissue, we had astrocytes, microglia, neurons, oligodendrocytes, and their precursors. The results of the differential abundance analysis revealed that women with the disease have more oligodendrocytes in nervous tissue and less natural killer cells in blood than men. Moreover, differential gene expression analysis show that every cell type has significant differences by sex. We integrated the later results afterwards. In detail, we identified for each dataset which genes were overexpressed in women and overexpressed in men. In some cases, we detected biological processes overrepresented. In addition, we seized the opportunity to integrate the results by cell type among the 2 blood datasets, for TCD4 cells and for TCD8 cells. Finally, some of the conclusions that can be extracted from this work are: first, describing sex-specific biomarkers in multiple sclerosis is key to approach the disease from the perspective of personalized medicine. Secondly, men have overrepresented functions related with antigen presentation in blood, while women functions involved in RNA processing in TCD4 cells and responses to different types of stresses in TCD8 cells. And finally, considering all cell types in the nervous system, we have detected the GO process sequestering of actin monomers enriched in men and 4 cell adhesion functions in women. That's it. Thank you for inviting me and thank you for your attention.